thanks for the invitation. This has been a great meeting. I'm really learning a lot, and it's so much fun to see people in person that I've seen only over Zoom, so this is great. So I'm going to talk about something that might be a little different from what we've heard so far. I come from NIST, National Institute of Standards and Technology. We're kind of an unusual organization. We're part of the federal government, part of the Department of Commerce, so that's a little different. We work a lot with industry because we're part of the Department of Commerce. That is our major constituent. And although we also, of course, work a lot with academics and other government agencies. But our major goal is what we call infrastructure for measurements. And we're particularly concerned with how does one evaluate the confidence that they have about the measurements that they make. So clearly measurements are critical for having confidence in medical devices, medical therapies, bridges. We have a very broad portfolio at NIST with a lot of mostly emphasis on the physical sciences and information sciences. And we also do actually participate in standards development, particularly documentary standards, but some of those are founded or supported by the basic research that we do in measurement technology that includes developing reference materials and benchmarking materials, reference data, et cetera. So we all know, how important data sharing is. One of the things that data sharing allows is, of course, more confidence, that if you know what I did, you have more confidence in what I did and what my results are about. But for me, the most important thing about data sharing in this space is that biology is really complicated. And if we cannot share our data with one another, we're never going to solve the major questions about complex systems like biology. So I think it's really, really important that, that we can share data so that that data can be analyzed further and theoretical models can be developed so that we can understand really how biology operates. And to me, that's the most important thing. One of the other important things, though, is, people talk about confidence in, or rather irreproducibility, and I always push back on that because I say, because we're not keeping track of exactly what we did, what I did in my experiment, or what you did in your experiment, we're probably most of the time doing different experiments. So we shouldn't expect that the results should be the same. So this is part of the challenge. I think that this community is addressing, whether it's part of your goals or not, you're addressing the challenge of how do we establish better confidence in our data. And of course, having all of this, having this confidence in data, being able to share data and therefore create new knowledge, requires a couple of things that are all parts of what this community has been talking about, which is really granular experimental information and analytical details. But I would also add to that indications of quality of the data. And by that I mean reporting uncertainties in data. That's not usually thought about in terms of data, but for me, the quality of the data is just as important as the volume of the data. And a lot of times that can come about through benchmarking and reference materials, can really help. Kind of the question is, compared to what is the way I always look at it. I have to apologize for being part of the US government and how poorly we have done. I'll just say that right out loud. Because I'm part of committees that are writing white papers, for example, on data for the bioeconomy. You can see that everything they say are things that you guys would agree with. But we've been having these conversations in the U.S. government for about 20 years or so, maybe 25, and we're still justifying why is it important to do, which is a little disconcerting. Now, one of the reasons this is a really important topic and a very timely topic is that optical microscopy has just exploded, fairly recently with the advent of automated methods, but also the fact that computational capabilities are so much cheaper now. And, you can really do a lot on a lot of data pretty fast, and it's not all that expensive. And then we also have access to better and better, I think, reference materials. I'm looking at the Argo slide kind of data, which is really, really handy. And so, we can develop lots of data and lots of quantitative data, but it's only going to be useful to us if we have the benchmarking information and if we have sufficient metadata. And so this metadata problem is something that I think is really eating people's lunch right now. And for very good reason. It's a really challenging topic area. And one of the things that's challenging that I see this community struggling with is what metadata to report. A lot of people want to think we can just report minimum metadata, whatever that means. And the way I think about metadata to report is from a metrology point of view, which is what are the variables in your experiment that you think are important to report? What are the things that might make your experiment result different from somebody else's experimental result? And so in metrology, we have this cute little thing called an Ishikawa diagram, or a cause and effect diagram. Sometimes it's also called a fishbone diagram. And it's just a systematic way of tracking your variables, what you think are important variables, and how you might mitigate them, and how those all add up to sources of variability. This is one that was created for an interlaboratory study, an international interlaboratory study on nanoparticle toxicity. But one could imagine setting this kind of structure up for, say, your microscopy instrumentation. And not everybody's going to have everything in their metadata, but for any experiment, you could certainly set up your own Ishikawa diagram for what's important about your your particular microscopy instrumentation. When it comes to biological biological components of an experiment, that gets really tricky. I mean, that's the part that keeps me up at night, because there are so many variables that are so fine detailed that are really, really, really hard to capture. And nobody's notebook captures them all. And even if they did, then they're not machine readable. So, it's really, this is really challenging. And this is an area that I would love to see more funding and more effort done to program, to develop programming tools that allow the biologist, makes it easier for the biologist to capture that information and have it in a digital form. So biological components are really tough. But then there's the analytical pipeline, which is also important to keep track of. And I'm not sure that I've heard a whole lot of information about that, or a whole lot of work on that. So I just wanted to provide some examples from our lab about how we would think about collecting such data and reporting such data. So we've been developing pipelines to image in phase contrast induced pluripotent stem cells and follow individual cells over time, be able to track them through divisions, for several divisions, for over a few days. And so we've created this pipeline with H2B GFP cells that have been produced by Allen Institute. Gives us really good ground truth for the nuclei, and also also for the mitotic cells, so that we've created some just segmentation and classification algorithms in order to follow cells that are in mitosis, cells that have divided, cells that are segmented, and so we can follow cells and their progeny over long periods of time. So this helps us do what I'll show in the next slide, which is why we're doing this. But what we were really interested in this project was how would we evaluate any pipeline that we created. People are creating AI pipelines all the time, right? But how do you evaluate those pipelines? And that was really what we were interested in. One of the reasons that we're doing this is we're interested in statistical thermodynamics models that give us a handle on the heterogeneity in cellular phenotype over populations. And that heterogeneity, we think, gives us a lot of information about how regulatory networks in cells work. But that involves looking at fluorescence fluctuations in individual cells on an intermittent basis, but over long periods of time. And so we really want to be able to recognize those cells in phase contrast so we can track them for long periods of time. So we asked a number of questions in terms of how to validate the pipeline that we have. We looked at several different models that were based on training data with different numbers of cells. One of the reasons that we wanted to do this, this automated segmentation process for our training data was so that we could sample lots and lots and lots of cells without having to do manual segmentation, because we want to collect all the heterogeneity that might be present in the population. So we tested different models. We tested how manual versus automated segmentation compared. We tested how well those different models worked when you were tracking them over long periods of time, influence of post-processing, thresholding, reproducibility. We looked at different cell lines. So we reported a lot of these results in a paper. The data for this is available at a NIST data site. Nothing fancy, doesn't have any metadata associated with it other than an Excel file that explains what the experiments were. They all track. We only published, put up the data that, that related to data that were in the published figures. So we end up with outstanding questions here, right? So, we did all this, we have this, but we don't really know how much data we should report. Or how much data to make available. I mean, these are all questions that we just fudged as we went along. And so, I look to this community to offer advice about, what really should we expect people to report and how much data should be available. I'm also really concerned about this idea about capturing and reusing experimental metadata terms. And I'm looking at some work that's going on at NIST in our AI, through our AI group, where they're trying to develop metadata for the CHIPS Act and using a lot of LLM approaches to create and gather data, metadata terms out of databases. And I look at that as maybe an approach that we can start thinking about in a community like this. And then I just want to acknowledge the people who worked on this. It takes a multidisciplinary team, which is one of the reasons that I've been at NIST as long as I have, is that we can really put this together easily. We have a biologist, biomedical engineer, high performance computing guy who came in as a postdoc and did most of the coding, and then a couple of people that we work with very closely from our information technology laboratory, and they're computer scientists. So that's it. Thank you.